Sciences, and we are doing a complete course on cell biology. We have discussed the basic definition of cell biology and history of cell biology in the last lectures. And in this video, we will further discuss the growth and development of cell biology in 19th century and as well as in 20th century. So this is the part 2 of history of cell biology. Earlier scientists were more focused on animal cells until 1807, when Mirbeau stated that plant tissues were also composed of cells. Another biologist, René Destroches, concluded that all animals and plant tissues were also composed of cells. In 1831, nucleus, which is the major component of the cell, was discovered and named by the English botanist Robert Brown. And the cells of epidermis, stigmas and pollen grains of the plant Tradescantia. He concluded that the nucleus was the fundamental and constant component of the cell. That means all the cells contain the nucleus. Cell theory, which was given by Schleiden and the Schwann scientists. Further, cell theory was elaborated by some more scientists. So there was a scientist like Schleiden and Schwann who suggested that cells are the basic unit of structure in the plants as well as in the animals. Schleiden, Matthias Jakob Schleiden, he was the first to describe the nucleoli. And he appreciated the fact that the cell leads to a double life, that is, one independent and another as an integral part of the multicellular plant. Another scientist, Theodor Schwann, was a zoologist, and he gave the same thesis and he applied these theses to the animals. He worked with the connective tissue such as bone and cartilage, and he modified the cell theory to include the idea that living things are composed of both cells and the products or secretions of the cells. He introduced the term metabolism. Metabolism is the term which is used to describe the activities of the cell. Whatever the functions of the cell takes place, we call it metabolism. They both postulated that the cell is the basic unit of structure and function in all life forms. Thus, simple, basic and formal biological generalization is known as cell theory. Cell theory was extended by K. Naegeli, and he said that plant cells arise from the division of pre-existing cells. Another person who was a pathologist, Rudolf Virchow, confirmed that the Naegeli's principle of cellular basis of life continuity, that means cell arise from the pre-existing cells, while when the pre-existing cells divide, they give rise to new cells, and that's how there is the existence of the cells. He also established the significance of cell division in the reproduction of organism. He published his textbook Cellular Pathology, and in that he concluded that functional unit of life, the cells, were the primary sites for disease and cancer. How can he say that? Because cells are the functional unit of life, and if there is any dysfunction in the cell, they cause diseases and cancer. Later in 1865, Louis Pasteur, he also gave experimental evidence of the virtuous extension of the cell theory. Now, the modern version of cell theory states that all living organisms are made up of one or more cells and cell products. All metabolic reactions in unicellular and multicellular organisms takes place in cells. Cells originate only from other cells. No cell originates spontaneously. There is no spontaneous origination of cell, but comes into being only by division or duplication of already existing cells. The smallest clearly defined unit of life is the cell. Here are some more points about cell theory. There were fundamental similarities in the chemical composition and metabolic activities of all cells, whether they are cells of plants or animals or any microorganism. There is a fundamental similarity in the chemical composition and the metabolic activities. Again, a scientist Colliker applied the cell theory to embryology. He demonstrated that organisms developed from the fusion of two cells. You can see in this video the spermatozoa and the ovum. And there was a study in the of the large number of subcellular structures. We can say subcellular structures to those structures which are present in the cell as a different unit. Like you can see in this diagram, you can see here there is nuclear membrane, there is nucleus, there is cell membrane, there are ribosomes, there is mitochondria, there is endoplasmic reticulum. These are units which are present in the cell which have their own functions to help in the metabolism of the cell. So each unit needs to be studied in detail, and so that we can study the whole program, whole metabolism of the cell. Now, there are some organisms present on the earth which are alive as cell but show some exceptional functions to the cell theory. So, cell theory does not have the universal application. That is, there are certain living organisms which do not have or are true cells. So all kinds of true cells have some basic characteristics, and these are some basic characteristics: a set of genes which constitute the blueprint for regulating cellular activities and making new cells. So what are genes? Genes are present in the cell to define how the cell, how the organism looks and functions. But these genes are transferred to the offspring of the organism or the cell. So these help in making the new cells, and these are present in the cells only. There is also a plasma membrane that permits controlled exchange of matter and energy with the external world. A cell is surrounded by this plasma membrane. There is a metabolic machinery which helps in sustaining all the life activities like growth, reproduction and repair of plants. This machinery should also be present in the cell. Now, there is an exception to this cell theory. These are viruses. Viruses do not easily fit in these parameters of a true cell. Thus, they lack a plasma membrane. They don't have a plasma membrane. They don't have a metabolic machinery for energy production and for synthesis of the protein. However, like any other cell organisms, viruses have a definite genetically determined macromolecular organization. There is an organization of macromolecules present in the viruses. You can see here, these are the diagrams of the viruses. Okay, there is a genetic and hereditary material. There is a present of genes. Genes are present in the form of DNA or RNA. Okay, a capacity of auto reproduction is there. So there is the capacity of reproduction, and there is a capacity of mutation also in the genetic substance. They don't have any metabolic machinery, they don't have a plasma membrane, but they have genes, they have hereditary material, they have macromolecular organization, they reproduce, and there is also a capacity of mutation in their genetic substance. So viruses can only reproduce inside the host cells. Host cells are animal, plants, or bacteria. Viruses use their own genetic program for reproduction, but rely on the raw materials and biosynthetic machinery of the host cells for their multiplication, like the proteins. The energy they need, they take from the host cell in which they live, and they use their own genetic program to reproduce. And they are defined as an infectious, subcellular and ultramicroscopic particle. We are saying here it is a particle representing an obligate cellular parasite. It is also known as a parasite, and a potential pathogen. It's a pathogen. Pathogen means the organisms which produce diseases, whose reproduction in the host cell and transmission by infection causes characteristic reaction in the host cells. So they show some characteristic reaction in the host cells. They are described as naked genes. That means there is no cellular formation, but genes are present there that had somehow acquired the ability to move from one cell to another. They transfer from one cell to another. Like that means they are infectious. They produce diseases from one cell to another cell, or as cellular forms that have degenerated through parasitism, or as primitive organisms that have not reached a cellular state. So you have read about the viruses, but there are also some organisms such as the protozoa, paramecium, the fungus rhizopus, and the alga vaucheria. They also do not fit in the purview of the cell theory. After cell theory, scientists came up with protoplasm theory. In 1835, a scientist named Felix Dujardin, he termed a jelly-like material within a protozoa as sarcod. It is a, we can say, cytoplasm, a cell sap which is present inside the plasma membrane, which is earlier it is called protoplasm, and now we termed it as cytoplasm. So in 1835, another scientist, H. von Moll, he described cell division, and further Czech biologist J. E. Purkinje coined the term protoplasm to describe the contents of cell. So whatever the contents present inside the cell, he termed it as protoplasm. Von Moll in 1846 applied the name protoplasm to the contents of embryonic cells of the plants. Max Kolche in 1861 established similarity between sarcod and protoplasm of animal and plant cells, and thus offering a theory which later on improved and called protoplasm theory by O. H. Hertwig. So uh, pr- protoplasm was termed by a scientist, which is the contents of the cell and of the plant, and after that it was uh, termed by 1 mole of the contents of the embryonic cells of the plants. And another scientist established a similarity between the sarcod and the protoplasm of both plant and animal cells, and later on protoplasm theory came up by the O. Hertwig. So next, the cell is an accumulation of living substance. This living substance we called it as protoplasm, which is limited by in a space by an outer membrane. We have an outer membrane around the cell, which is we can say plasma membrane, and cell contains the protoplasm and possesses a nucleus. The protoplasm which is filled in the nucleus is called nucleoplasm, and that exists between the nucleus and the plasma membrane is known as the cytoplasm. The last quarter of the 19th century is usually considered as a classical period for cell biology, since various significant cell biological discoveries have been made during this period. So, in 20th century, there was a great advancement in cell biology. Why? Because of the two reasons. There was an increase in the environment instrumental analysis, because there was an introduction of the electron microscopy and X-ray diffraction techniques. If you want to, we will discuss these techniques in the further videos. Okay. So there was also the convergence of the cytology with other fields of the biological research like genetics, physiology, and biochemistry. So because of this advancement in techniques, there was uh, there was a development in the detection of the molecular components of the cell, and other cellular components have been separated by the other techniques like ultracentrifugation. Different biochemical events of the cell was also studied by autoradiography, and methods of tissue culture also developed that time. That's why we can also study the living cells, right? Then phase contrast microscopy and interference microscopy came up, which helps in the study of the living cells. The structure of the cellular membrane, it can be observed by the techniques like freeze fracturing and the freeze etching. Micromanipulators, micromanometric methods, and chromatography, electrophoresis, spectrophotometry. These are the techniques which help in the investigation of the details of the cell and its components. Because of these employment of various techniques in the study of the cell, the cell theory and the protoplasm theory, they also became more advanced and more uh, become popular, and people can also study more about organism, and that's why the theories came about. A new theory which is called organismal theory. Here organismal theory is presented here, where the body of all multicellular organisms was studied, and they were called as a continuous mass of protoplasm. And the protoplasm is divided into incompletely into small centers, which we call the cells, and for various biological activities. So multicellular organism is a highly differentiated protoplasmic individual. If you see yourself, you are a multicellular organism. So when the organismal theory came up, they says that an multicellular organism is a continuous mass of protoplasm which is divided into small segments which we call the cells, right? Then unicellular organisms like protozoa, they are small in size, and there was small degree of differentiation of the protoplasm. The differentiation involves separation of the protoplasm into subordinate semi-independent compartments we call cells. Even the embryological development of the multicellular organism include only growth and progressive internal differentiation of a small single protoplasmic individual, which is egg. Organismal theory but fails to ascertain the position of viruses. It cannot position the viruses. What are viruses? And we have talked about that it is the exceptional to the cell theory, and we will further discuss more about viruses in our upcoming videos. So thank you very much for watching this video. I hope you understood the concept. I hope you like this video. If you like it, please press the thumbs up button and share this video to your friends and family members who really need to understand this concept, and subscribe to this channel for more upcoming videos and press the bell icons for more notifications. Thank you.